All right, next I'd like to invite Dr. Wesseling, who is a consultant breast pathologist at the Netherlands Cancer Institute. His work is very well known to this audience, but his research focus has been to develop new strategies to improve the prognostic and predictive abilities of breast cancer pathology. I've been very fortunate to work with him in his interests in DCIS, and he is the lead PI for a multi-center international collaboration funded by the Cancer Research UK Grand Challenge Award, which focuses on trying to determine risk for patients with DCIS. So he will talk to us today about clonal evolution of DCIS to invasion. Welcome, Dr. Wesseling.
Thank you so much for the introduction, and I would like to thank the organizers for inviting me to share our results. Also on behalf of the team, of course, our results with you, because these are, the scientific results to be shared with the community and society. And I talk about the clonal evolution of DCIS, what we know about this, but also beyond the clonal evolution of DCIS. So here are my disclosures. The thing is, the issue with DCIS is that DCIS at the time of diagnosis, the ones that do progress and the ones that will never progress, they are often lookalikes. And ideally, you would like to distinguish them at the time of diagnosis between the indolent ones and the ones that progress to invasive disease. However, this is not what we can do right now, and due to this uncertainty, we treat them almost all. There are three interrelated questions that we need to answer. First of all, and I think the previous talk already was dealing with this as well, what drives or prevents progression of DCIS to invasive breast cancer? The second question is, how can we distinguish indolent from potentially progressive DCIS? And the third question is, the subsequent invasive breast cancer, is it really related to the initial primary DCIS lesions? And I start with this last question. And I refer to the paper that was published this year on behalf of the Precision team, where we compared 95 pairs primary DCIS lesions with a subsequent ipsilateral invasive breast cancer. And these occurred 1 to 70 years after the initial diagnosis. And we did that on various levels. Whole exome sequencing, panel sequencing, copy number alteration analysis, and for a few of a small number also single cell analysis. And I show you some of these results. Here you see, for example, on the whole group, the copy number alteration analysis, and then you can appreciate that the copy number alterations are quite similar between the initial primary DCIS lesions versus the ones that come back as invasive breast cancer. Only for 1q and 3p you see more often a gain in the invasive breast cancers, and you see a lower incidence of loss of at 3p in the DCIS lesions. And also the usual suspects are there, both in the DCIS lesions at the left side versus the ones at the right side, the subsequent invasive breast cancers. And when we look then at individual cases, that was also apparent already from some mutation panel sequencing results and copy number alteration analysis, you see that there are quite a few, the majority is really clonally related, as you can appreciate from the top three panels, these scatter plots with the variant allele frequencies. Whether the independent ones, they do not share any mutations, as you can see below. And just looking at the single cell sequencing that was done on FFPE material, old FFPE material in the lab of Nick Nevin, then you could appreciate, you could see that the unrelated really do not share, I do not have a pointer here, right? That the unrelated ones, they do not share any clone, as you can see in this Muller plot and this Hume plots, whereas the related ones, they really share mutations, clones. So then it comes to the final score, the clonality score or class, if you like. And what is striking from this one is that, at least in this series, you don't see a relation to the ER status, to the HER2 status, and to the time to recurrence. And that 1 out of 5 appears to be a brand new invasive breast cancer, not related to your initial DCIS lesion. How can we explain this? Well, first of all, one might think of these clonality models that has been published before. We found out multiclonal invasion, yes, it's out there. Independent lineage progression to invasion, so a new primary. It has also been published, for example, by the group of the team of George Rice-Filio in 2017, that there are also cases where just a single clone or a minority of clones is invading. So these are the models. So what do we know from these results thus far? Well, genomic evolution occurs at very early stages of progression in ducts. Most genomic changes are already there. And DCS is both a precursor as well a risk lesion to invasive breast cancer, a potential precursor, I have to say. Clonal relatedness is not necessarily related to time to recurrence, grade, or subtype. And the risk of invasive breast cancer is likely overestimated once we realize that 1 out of 5 is a brand new lesion. That also makes clear why 0.5 is so important, that it impacts the prognostic value or power of prognostic biomarkers in DCIS, as 1 out of 5 is just a new lesion. So you have to overcome, so to speak, the noise that is in there, because also new lesions can occur. What then can explain the unrelated invasive breast cancer, the increased risk for invasive breast cancer? Is it a genetic predisposition? Is it a field effect? Or is it the microenvironment? Or is it a combination thereof? Therefore, it's very important to realize that how the mammary gland comes in play over the years, during lifetime. We know that mutations in the mammary gland already can occur very early in life, and that they are propagated, as is shown in these pictures, using a confetti construct over an entire segment, once this mutation occurs very early in life during embryogenesis, for example, putting this segment at risk, a so-called sick lobe, generating a sick lobe. And schematically simplified drawn, you see that over here as well, where you have a rudimentary gland where mutations can propagate over an entire tree. And this can happen also during puberty, when there is extension and branching and formation of terminal end buds, etc., where a subsegment can contain such a mutation. And during adulthood as well, during the menstrual cycle, for example, where remodeling takes place, etc., etc. So this could be a contribution to the risk of subsequent invasive breast cancer once a DCIS lesion is there. And it's also something that you see in mouse models that are deficient or mutated for BRCA1 and P53, where you see that these cells all are deficient for BRCA1 and P53, not necessarily transformed to invasive breast cancer in this case. Some of them do, the green color, so the green mammary stem cells, so to speak, they transform to breast cancer. But for the red segments, so to speak, they do that only in part, whereas there are also segments that do not transform. So there's something else at stake that we're probably not aware of. Well, maybe Rob was already alluding to this a bit in the previous talk. And therefore we have to realize that what we can learn from 3D tissue analysis, because the human mammary gland is of course a 3D organ with different segments, with sick lobes in it, yes or no, and normally we only look at it in 2D, a kind of snapshot pathology, I would say. And therefore we developed a technology that was mainly done by Hendrik Messal, where we can make breast tissue transparent, apply antibodies to this, and scan these 3D tissue specimens, even whole breasts, and study this in more detail. And what you already see in a normal cosmetic reduction is a number of abnormalities. This is from a premenopausal woman. Hypobranched areas, also normal areas, fortunately, dilated areas, areas with hyperplasia, but also areas with DCIS in some cases. And the burden of these deformations increase with age, as you can see in this graph. So there's all kind of stuff going on that might relate to a field effect, that might relate also to a genomic predisposition that we're not fully aware of because we didn't discover it yet. And when you dive into this a bit more detail, you even see in one cosmetic reduction 4 different trees where you see these hypobranching or this branching ducts and these dilations, etc., are present in the aging breast, I would say. And if such a breast, this is an example of a preventive ablative specimen where you see where we found DCIS by accident, and you see in the very same segment at different places, you see these DCIS lesions with a similar morphology, but no connection between these DCIS lesions. And how does it relate to a 3D movie? I could not show the movie here, so I made pictures for this, where you have a part of the breast tissue. This is from a FFPE block with normal ducts and TDLUs at the left side and DCS at the right side. Now there are 2 striking findings, or maybe 3. The first striking finding that I was not aware of, and that we are checking right now, is that there are no lobuli anymore in the area where there is DCIS. The second is that the ducts are extended, that's no surprise. And the third striking finding is that the ducts sometimes lose their continuity. As you can see in the lower right picture, where you see that the ducts somehow are not continuous anymore with the whole ductal system. What it means, I have no clue, but it might be that it also relates to both genomic predisposition and the field effect. Can we then sequence the DCIS and the adjacent normal-looking ducts? And for this, also the Flash 3D technology was applied, where you can sequence in 3D, so to speak, the DCIS lesion in red and the adjacent normal-looking ducts. And what we show over here is that indeed in the normal-looking ducts, you see also already some copy number alterations that are also present in your DCIS lesion. And at the very right-hand side, you see a reconstruction of your ductal tree in red and the normal-looking ducts in green. So what can we learn from this? Well, that there is a high burden of benign tissue deformations in disease-free breasts, and that tissue remodeling can clear a lot of mutant cells, so a lot of mutations as well, but it can also boost expansion of remaining mutant cells, as I showed you for the mouse model, for example. And these mutant cells can spread mutations over large areas without causing cancer. Think about the sick lobe syndrome. And DCIS and normal neighbor ducts share genetic alterations. And keeping this in mind, we can think about the microenvironment. Is this driving then the expression in the luminal cells of the DCIS lesion that really comes with progression to invasive breast cancer? And can we see that then in our RNA expression signature of these DCIS cells? And this is my last slide for this. Probably yes, because we do not see a clear difference in mutations that are out there or copy number alterations, but there is a clear difference in RNA signature that we find in our case controls series regarding the RNA expression signature. So what is then our hypothesis? Well, you have a mutant cell and this is repressed or even it disappears over time. And that cell can be transformed to a malignant cell once this maybe active repression is released by genomic alterations combined with microenvironmental changes. and then leads to initiation of a tumor. And with that, I would like to stop, but first I would like to thank our patient advocates who are they're also here again. So thank you so much. They're writing plain language summaries. They elicit a lot of discussions that are very helpful. I would like to thank the whole Precision team. So forgive me if I forget one name to mention here. But this picture was from our last in-person meeting in Amsterdam that was finally possible again. So thank you for your attention. I'm happy to take any question.
Thank you very much, Dr. Wesseling. I think we have time for one question. The mic over there, please.
Steve Nayrod, very interesting. I think, though, any model of DCIS to invasion should have to consider contralateral breast cancer as well. The risk of contralateral breast cancer following a diagnosis of DCIS is the same as that of following a diagnosis of invasive cancer, 0.4% a year for the rest of the lifetime, which is approximately the risk of the unrelated invasive cancers in the ipsilateral breast. So I think if you consider a fifth of the ipsilaterals are new, de novo, unrelated, with the same exact risk of contralateral invasive cancer in both breasts, I don't think a model would really work if it's only focused on the ipsilateral side. Should consider that both breasts become predisposed at the same time.
Yeah, this is a very interesting question for a number of reasons, but because of time I will limit this to two reasons. One reason is the genetic predisposition issue that I talked about. The other reason is that the microenvironmental issues. So once, for example, a woman has dense breasts, we always talk about dense breasts. So these are two. So that might play a role there as well in the enhanced risk contralateral as well. And that's also the message here is that we should be aware of the fact that a subsequent invasive breast cancer can be totally unrelated, but is initiated in a background of a predisposition, whether that's a field effect, whether that's the collagen, whether that's whatever that is out there, whether that's the genomic predisposition, that we don't know.
Well, a lot more work that we need to do to
Sure.
figure this out. Thank you so much, Dr. Wesseling.